our speaker, Rachel Owen.
Thank you. Okay, so I'm a third-year PhD student at Southampton University, working in Hannah's lab, who's just behind you. And yeah, my name's Rachel, and I'm going to be talking about my PhD project, which is on using the cellular origins of contagious cancers to try and identify vaccine targets. So just a bit of a background on cancer in general. So cancer is a group of diseases that are caused by uncontrolled cell division. So you get a healthy cell which acquires a mutation and it becomes cancerous. And this cancer cell then continues to divide uncontrollably and potentially forever. And that's how a tumour forms. But for a tumour to actually form and survive, it has to overcome quite a few different cellular defences. So these include things like the immune system, which is constantly fighting the growth of tumours, mechanisms in place to control the cell cycle, and mechanisms to actually stop the growth of cells, and also mechanisms which are in place to force cell death. So in order for a tumour to actually grow, it needs to overcome all of these. And this kind of opens up the idea of the hallmarks of cancer. So these are a set of things that cancers need to be able to do in order to actually form and grow and survive. And I mean, there's quite a few things. It's mainly avoiding the immune system, sustaining proliferative signalling, being able to avoid growth suppression, avoid cell death. But the one I'm really going to be focusing on is the idea that they have to evade the immune system. And this is a particularly interesting one to look at in terms of contagious cancers, which is what I'm really going to be talking about. So these are something people usually haven't heard of. And so I really want to ask the question, how does a cancer actually become contagious? So there needs to be a few things in place for this to happen. First off, the cells have to have a physical route of being able to transmit between two different individuals. The cells that do transmit then have to be able to invade tissue, and the cells have to be able to divide, and they have to be able to grow like a cancer would. Which brings in, they also have to be able to avoid the immune system, and specifically the anti-tumour response of the immune system. And I guess most importantly for a contagious cancer, they have to be able to evade the anti-graft response, which is something most cancers don't have to come up against. And there are at least 8 incidences in nature where cancers have gained the ability to be contagious, to move between different animals. And these are what I'm really going to be focusing on. So, one of these is Canine transmissible venereal tumour. So this is a sexually transmitted tumour that circulates in dogs and other canines, so wolves, foxes, coyotes. And this is really widespread. It's across pretty much the entire world. And actually the UK is one of very few places that doesn't have this cancer. It's predominantly associated with countries that have high levels of stray dogs, or countries like the USA where they kind of just let pet dogs run around free. Because we don't have that, we've actually managed to avoid this tumour. And what's interesting about this tumour is that generally it's not fatal. So they spread and grow really, really fast, quite quickly. But then there comes a point where the tumours suddenly stop growing, and often they completely disappear, and the dog is fine, and very rarely do they even need treatment. So this was first identified in the late 1800s by a vet who noticed it was transmissible. But actually they think this cancer is probably about 10,000 years old, and probably emerged in a wolf or one of the really ancient dog breeds. So this cancer has been going for quite some time now. So there are 5 of these cancers actually circulating in 4 different species of marine bivalve, so mussels and clams and cockles. These were described over the last 3 years, where they've actually studied them and realised that they're contagious. But again, we know these cancers have been around for quite a bit longer, so it's only recently that we've started to understand them. And what's interesting about these ones is that at least one of them has actually crossed the barrier between species. England have scored. Yeah. So one of these cancers formed in one species, but now it only exists in a different species. So it's managed to seriously avoid the immune system there. And the fact that we have 5 of these circulating in different species of clams kind of suggests that maybe these transmissible tumours are quite common in the marine environment. Before I get on to the final two, I just want to talk about examples in humans. So we don't really have any true examples of transmissible cancers in humans. We don't really have the routes of transmission that they need, and our immune systems are pretty good. But there are rare incidences where actually cancers do transmit between people. So one of these is during organ transplant. You can get donor to recipient transmission. It's very rare, and this is actually usually because the recipient is on immunosuppressants. So the immune system is dampened, which means these cancers have a better environment to grow. Another example is mother-to-baby transmission to the fetus during pregnancy. And again, this is even rarer. It hardly ever happens. But again, the fetus has a level of immune privilege to stop the mother's body from rejecting it as foreign. So these only really happen in cases where the immune system is dampened in some way. Probably the most interesting example is this one. So the tapeworm that turned into a tumour. This was a man in Colombia with quite late stage AIDS, so he was quite sick, had quite a bad immune system at this point. But they found he had lots of strange nodules in his lungs and they didn't know what they were. When he died, they took some biopsies and it turned out he had a massive tapeworm in his gut that had developed a cancer, and the nodules in his lungs were actually cancer from the tapeworm, which had spread into his body and were able to grow there. So, I mean, cancers can be quite amazing in what they're able to do. But again, this is a case of serious immune suppression in the recipient. So these don't really happen in humans that often. Now the final two true contagious cancers I'm going to talk about, and these are the ones I'm really focused on today, are devil facial tumours. So these are two different tumours that circulate in the Tasmanian devil, called DFT1 and DFT2. So they both cause these really nasty, necrotic, rotten tumours around the face and mouth, which have a really high mortality rate. They kill the devils most of the time. And they're spread during biting. And it's interesting because this is the only mammal species that we know about that harbours two different contagious cancers. And these are really what we focus on. So, a bit of background to the Tasmanian devil, which a lot of people don't even realise they actually exist. So this is the Tasmanian devil. This is quite a young one, it's about a year old, and yeah, they're sweet little things. So these are the world's largest carnivorous marsupial, and they're endemic to the Australian island of Tasmania. They used to live on the mainland as well, but they went extinct on the mainland about 400 years ago after humans settled and started killing them off. So now they only live on Tasmania. So like all marsupials, they have a pouch. They give birth to their young very, very underdeveloped. So for the first 4 months of the joey's life, it lives in the pouch where it does most of its development. But once they're fully developed, they actually become independent very quickly. And these marsupials are generally solitary and they're nocturnal. So you might be wondering how such a cute looking animal got the name the Tasmanian devil. So I have here give it a little change, just a short video which should explain that. devil. So their name actually originated from when people first settled on the island. These animals are nocturnal, so they very rarely saw them, but they would hear them feeding. And I think you can see, if you heard that coming out of the darkness in the middle of the night and you didn't know what it was, it would be, you'd think it's quite demonic. So, and these feeding frenzies that they have when they all come across a carcass involve this loud screeching and growling and a lot of fighting, and you probably saw them snapping at each other a lot. They bite each other, and these animals have one of the strongest bites in the animal kingdom relative to their size. So a bite from, you can see one eating bones. A bite from one of these can cause a lot of damage. So these guys damage each other's faces a lot during these feeding frenzies. Because of kind of these two things mixed together, the way they sound, their aggression towards each other, these guys have been historically persecuted by farmers who thought that maybe they were hunting their livestock. So they've suffered quite a lot at the hands of people. Their population has been decimated a few times by us. In reality, they actually don't hunt that often. They tend to go for roadkill, things that are already dead. They're primarily scavengers. And actually, farmers are now realising they're quite beneficial because they clear up a lot of the dead stuff and they stop infection from spreading to their animals. So, despite this persecution, historically they've been abundant on the island. There have been lots of Tasmanian devils on Tasmania for a long time. But despite, they sometimes got killed off and their numbers dropped, but they always came back. But since '96, the populations have been in really, really steep decline. So overall, it's been over 60% drop in animal numbers now. But in some parts of the island, it's as high as 90% of the animals have been killed off. And as of 2008, they were reclassified from least concerned to endangered by the IUCN Red List. So this is now an endangered species. And this decline is pretty much entirely due to the emergence of these devil facial tumours, and in particular DFT1. So these probably look a little complicated at first. Essentially what we're looking at is the number of devil sightings in a 10-kilometre area and years. And what you can see, these pink arrows show when DFT1 first emerged, and the populations of the number of devil sightings in these areas have been dropping steadily ever since DFT1 arose. So this cancer has really caused severe problems for the Tasmanian devil. So I'm going to be talking now about devil facial tumours, which is what we work on. So how have these tumours emerged? The short answer is we aren't really sure. There's no evidence of any external causes. These don't seem to have been caused by UV, they don't seem to have been caused by external pollutants, so we really don't know why they're appearing. The fact that they've been historically persecuted and killed off quite a few times has really reduced the genetic diversity of the species, which can reduce the species' health and make them really prone to suffering from diseases. So that's a risk factor. And the fact that they bite each other so often and they are constantly wounding each other is also a risk factor for transmissible cancers like this. But we don't actually know why they're appearing. So DFT1 was first identified in this northeast corner of Tasmania in 1996, but we're not sure how much earlier it actually arose. Nowadays, it exists across pretty much the entire island. It's spread quite massively. They did recently find a population down here in the southwest that is currently disease-free, and they're going to be doing some genetics on them to see if there's actually a reason that they're disease-free or if it's just not reached them yet. But generally, most of the island has this cancer spreading in the devil populations. So DFT1 emerged in something called a Schwann cell. Now these are a cell that exists in the peripheral nervous system, so not in your brain, but in all the other nerves in your body. And what they do is they wrap around the nerve axons, and they form something called a myelin sheath, which is just a fatty layer that speeds up nerve conduction. And only two types of cell actually produce this myelin sheath, and one of them is a Schwann cell, and the other one is the equivalent of a Schwann cell in your brain, called an oligodendrocyte. And a specific protein for Schwann cells, periaxin, and you don't need to know what it does, but it's a marker for DFT1, which was the first evidence that that's where DFT1 came from. So DFT2 emerged in 2014 in this southeast region of the island. And we can't find evidence of it existing much earlier than that, so we think it literally emerged about 4 or 5 years ago, and it's quite a young cancer. And it has currently yet to leave this region. So it's only been officially documented in 11 devils, but it's got about a 10% disease prevalence in that region of Tasmania. And we don't know where DFT2 came from just yet. What we do know is, despite the fact that they look the same, these two tumours are different. They arose independently. So these are just slices of tumour tissue, and the darker purple dots are the actual tumour cells, and the pink stuff is just stuff around the tumours. And what you can see is DFT1 here, the tumour cells are in these nests, bundles, and we see that in all DFT1 tumours, whereas DFT2 doesn't have this. Its tumour architecture is completely different. And also, so I mentioned periaxin was a marker for DFT1. DFT2 is completely negative. So this brown staining is periaxin positive, and we see none of this in the DFT2 tumour. These two brown bits down at the bottom, they are nerve bundles, so that's Schwann cells that are staining for periaxin there. The tumour itself is negative. Also, this looks a bit complicated, so these are all the chromosomes that we see in a normal, healthy male devil, DFT1 and DFT2. And all I want to point out to you is that DFT1 has these 4 marker chromosomes, which we see in all DFT1 tumours, and we never see in healthy tissue. That's something that's been caused by the cancer. And I also want to point out that DFT2 has a Y chromosome. So DFT2 has emerged in a male devil. We can't find any evidence of a Y chromosome in DFT1, so it looks like it's emerged in a female devil. So these two have completely different origins. They've emerged separately, and they are separate tumours, despite the fact that they look the same and they spread the same way. So people find contagious cancers quite surprising, because cancer is not normally a contagious disease. And this is because our immune systems can recognise and remove foreign cells. So if you have 2 mice here that are different, if you try and do a spin graft from one to the other, you get rapid graft rejection. The graft that you try and move over will die because the immune system of the recipient mouse recognises it's foreign and kills it. And this is because of something called the Mhc Class 1, which is essentially a cellular barcode. It consists of these 3 elements: a heavy chain, which is very variable within a species, a small accessory protein, beta-2M, and this peptide at the top. So the peptide is just a fragment of a protein, a short fragment, 8 to 12 amino acids long, and this is just generated as the proteins in your cell are broken down and reformed. So normal protein turnover, some of them just get put on the cell surface as part of Mhc Class 1. So this complex, it forms together, sits on the cell surface, and it acts as a barcode to the immune system, giving a snapshot of how healthy the cell is. And the immune system can then scan these molecules, and it can look at the heavy chain and the peptide, and if either of them are foreign, it will destroy the cell. So we're in a situation where, we have 2 genetically different mice. What's most likely different about them is this heavy chain. And the immune system notices that really quickly, and we get rapid graft rejection. Now, the immune system can also recognise the peptide. So if you have 2 mice, which are genetically different, but they have the same Mhc Class 1 heavy chain, but the peptide is different, the immune system can still notice this, and it will reject the graft. But it will reject it much slower. The only situation where grafts can actually survive between 2 individuals without immune suppression is if they're genetically identical. So your Mhc Class 1 is the same, and the peptide bound to it is the same. And if that happens, the graft will survive. And so why don't devils reject DFTs? They have a functional immune system, and we know they can reject skin grafts. There's been experiments done on this, and they reject them quite quickly. So they should be, these contagious cancers are essentially cellular grafts. They should be rejected. So we don't know why they're not doing it. What has been found out recently, and I will expand on this a little bit later, DFT1 doesn't express Mhc Class 1 on its cell surface. So to hide from the immune system, it's just got rid of the barcode. So there's nothing for the immune system to scan and find as foreign. And DFT2 does express it, so we're not entirely sure how it's avoiding immune detection just yet. But the fact that DFT1 is removing it, and the fact that it's there and devils can reject skin grafts, makes us want to see, is there any way to actually exploit this system and the fact that devils can recognise foreign cells to see if we can actually start it recognising DFTs. And so our lab is really working on trying to reduce the impact of DFTs, and specifically through something called a peptide vaccine. But before I get into what we've actually done, I'm just going to talk a bit about how we do it. So we get asked a lot about what samples we use, because obviously we're in Southampton, not Tasmania. So how do we do this work? Well, we predominantly work in cell lines. So essentially what happens, if you get a tumour, there's cells growing in it, and you can actually smash the tumour up and isolate some of those cells. And we can then, if we put them in the media, which is just sugar and nutrients and things the cells need, a flask like this, the cells will just stick to the bottom and grow. So these are DFT2 cells growing in a flask. That's what they look like. And the good thing about these is that we get sent them once, and then we can grow as many as we want. So we can grow them up, we can freeze them down, we can grow them back up again, we can get billions, we can have thousands. We can do what we want with these, and they're essentially a limitless source of biological material for us. But there are limitations. So obviously this is not how the tumours grow in the wild. So there's a lot of debate about how well these cells really reflect what the wild cancer looks like and acts like. So on top of this, we also used fixed tumour samples. So I showed you those slices of tumour earlier. That's a fixed tumour sample. So they're fixed in you take a biopsy from a cancer in a wild devil, you fix it just to make sure everything stays in the same place, and then you can embed it in wax and cut it to look at slices of it, or you can smash it all up and look at all the genes it's expressing. But obviously, these need to come from somewhere. So no one keeps devils in the lab. They're an endangered species. I don't think people would be too happy with us doing that. So all of this has actually initially come from wild devils. That means we need to catch them first, which is always good fun. So they are nocturnal and shy, which makes them quite hard to catch. And there are a lot of problems with, we often can't catch the same devil twice. So statistical significance when you're working on wild devils is a real problem, which is why we tend to stick to cell lines, because we can grow up as much as we want. But we have collaborators in Tasmania who perform regular trapping trips. So they take these white bucket-looking things out into field sites that they have, where they know there's devils. They'll put them in set places overnight, come back in the morning, you see if there's any devils in there. And if there are, they can be, they'll be visually assessed to see if they have any signs of the tumour around their face. If they do, we can take biopsies, we can take cells. Sometimes if the tumours are really bad, the devils will be euthanised rather than if their quality of life is going to be really affected. But most of them are released, and this is actually my supervisor Hannah, releasing a devil from a trap. I've still not got to go out and do this. I'm hoping to get to before the end of my PhD, but we'll see. But this is where all of our samples come from. So they do initially come from these wild devils. So what do we do with them? Well, so we have a situation where we've had 2 contagious cancers emerge in one species in just over 20 years. So that kind of suggests there might be a risk of them developing more of these. And the fact that they have such a reduced genetic diversity means that the species' health is not very good. So if more of them emerge, they're potentially going to cause more damage to the devils that are left. So it's really quite urgent that we start finding ways to help them. So there's a lot of work going into trying to support these populations. So in particular, there's quite a lot of work into breeding and re-release. So one of the small islands on the outskirts of Tasmania has been populated with healthy devils, which are breeding, genetically diverse, which are being regularly translocated back to the island. This is the Maria Island Translocation Project. So this is to keep the genetic diversity on the island high and try and keep the population up. We also have zoos that are breeding them in captivity and re-releasing them. But molecular biologists like us are generally nowadays trying to work towards some sort of vaccine. But as I mentioned earlier, we're particularly interested in Mhc Class 1 and whether we can use this for a vaccine. So before we can do that, we have to really understand what Mhc Class 1 is doing in DFTs. So I'm going to essentially sum up about 6 years of work in one slide here, but I'll do my best. So I mentioned DFT1 doesn't express surface Mhc Class 1. So this is flow cytometry looking at beta-2M expression on the surface. So it's Mhc Class 1 expression on the outside of the cancer cells. This is cell count on the y-axis and expression is on the x-axis. So this dotted line, this is negative beta-2M, and the pink line is DFT1 staying for beta-2M. So it's negative. There's not expressing Mhc Class 1. And again, I mentioned DFT2 does express Mhc Class 1. So we see this shift where DFT2 is expressing beta-2M on its surface. So that was shown by another PhD student in our lab last year. Now, this is where it gets slightly interesting. If you treat them with an inflammatory cytokine, interferon gamma, which just causes inflammation and it kind of makes the cell go into an immune response, we see an upregulation in surface Mhc Class 1. So if you inflame the cells, they suddenly start expressing Mhc Class 1. And even though DFT2 already expresses Mhc Class 1, I have recently shown that it also upregulates Mhc Class 1 if you treat it with an inflammatory cytokine. What's interesting here is these are just 3 different DFT2 cell lines. So this is all DFT2 and this is DFT1. And essentially what we find is that actually DFT2 upregulates Mhc Class 1 significantly more than DFT1 does. So it brings me back to, can we vaccinate against DFTs? Well, there's been a small number of wild devils, so these have just been tracked and tested, that actually show an immune response to DFT1. And this is correlated with progression of the tumours, and actually some devils survived for up to 3 years then with no evidence of the disease returning. So that's really, really good news. But these devils had in their blood antibodies against Mhc Class 1 positive DFT1. So the Mhc Class 1 on the DFT1 cells is eliciting an immune response. And if you take these Mhc Class 1 positive cells, you can actually vaccinate against DFT1. And there's been some success in doing this, but at the moment it's quite limited and the responses are a bit inconsistent. So this is really what we want to try and refine. Can we use these Mhc Class 1, or specifically their peptides, to actually vaccinate against DFTs? So it's clear that Mhc Class 1 is definitely involved in the immune response to DFT1, and its presence on DFT1 means that the devil's immune system can clear out the cells a lot better. And the fact that it's upregulated in DFT2 suggests that maybe it's relevant in DFT2 as well, and this is what we want to look at. So a peptide vaccine could be feasible. Now, these are something being really extensively explored for use in human cancer, but they're very complicated. So the basics of it is you have a healthy cell. It's expressing a normal peptide on its cell surface, and the immune system doesn't bother about this. It doesn't flag anything up. When it becomes a cancer, there's a mutation which messes up a lot of the proteins in the cell. If just one of these proteins happens to be expressing a peptide on Mhc Class 1, suddenly we have a new peptide on the cell surface, which is potentially foreign to the immune system. And if we can use that as a vaccine target, maybe we can elicit an immune response that only affects the cancer cell. So in theory, this is tumour specific, so there shouldn't be any adverse side effects, which is what we usually see in cancer treatment. So this is a really attractive option, but they're difficult. So this is just an example of a study. So they compared healthy mouse genes with mouse cancer genes, and what they found is that even though you have thousands and thousands of mutations in the DNA, very few of these actually become tumour-specific antigens. So very few of them are on the Mhc Class 1. And even fewer of them actually have any response from the immune system. And even if you get to that point, you can have similar peptides on other cells which will cause a response to these. So you can have off-target effects that have actually been fatal in some clinical trials. So this is a really difficult area to work in. And before we can even start on vaccines, we need to really, really understand what peptides we're actually seeing on these cancers. So how do we find these mutated peptides? Well, in a single organism cancer, this is reasonably easy. We have this, our organism forms a cancer, so we go back to the organism that's formed it, we isolate the cell type that the cancer mutated from, its healthy cell type of origin, and we make a direct comparison. Because if you can compare all the peptides, you can see this one is mutated. Great. So it's a bit different in DFTs. So we've had two Tasmanian devils, which have both undergone independent mutagenic events to form DFT1 and DFT2. Both of these devils are dead. They will have died either because of the disease or just because they're old. Devils don't live very long. So we can't make these direct comparisons. What we have to do instead is look at the current healthy Tasmanian devil population, isolate the healthy cell type of origin from them, pool all of that together, and then we can compare this to the cancers and try and identify specific mutations. Now, we can do this for DFT1 because we know it came from a Schwann cell, but we don't know where DFT2 came from yet. So this has been a huge part of my project, which is what cell type did DFT2 originate from? And so I'm going to give you a very whistle-stop tour of 2.5 years here. These look a little complicated. Essentially, this is the top 20 proteins that are overexpressed in DFT2 compared to fibroblasts, which are a healthy control, and DFT1. And all you really need to know is that every protein that's in red is a protein with a direct function within the nervous system, and every protein in blue is a protein that we see overexpressed in the nervous system. And if you just look at the colours, what you can automatically see is that DFT2 is really enriched compared to fibroblasts. It overexpresses lots of proteins from the nervous system. So it looks like a nervous system protein. Now, if you look at gene expression, so these are RT-PCRs. This is a way of looking at the genes expressed in cells. So these are DFT2, these are DFT1, and we have fibroblasts over here. And it's all compared to a control, which shouldn't change between cell lines. And all you really need to take from this is that these are all myelin-associated genes, which we should only see in 2 cell types. And we're seeing them all in DFT2. At a lower level than in DFT1, but they're there. Which means DFT2 has come from a cell type that produces myelin. And when you stain DFT2 tumour sections for Schwann cell markers, MBP and S100, and this is sciatic nerve as a positive control, which is really highly enriched for Schwann cells. What you can see is there is brown staining on DFT1 and DFT2 for both of these. So DFT2 actual tumours are positive for Schwann cell markers. So my conclusion really is that DFT2 has emerged in a similar cell type to DFT1. It produces myelin. It's from a myelin producing cell type, which is either, it brings it down to 2 cells. It's either a Schwann cell or it's an oligodendrocyte. And, but we think it's originated in a more immature cell version of this than DFT1 did. So this suggests that maybe devil myelinating cells are predisposed to contagious cancers. So it kind of really highlights how important our vaccine strategies are. So we're now in a situation where we have 2 independent cancers which have both emerged from similar cell types. We know that if we treat these cancers with interferon gamma, we can upregulate the amount of Mhc Class 1 on the cell surface, which means we're putting more peptides on the cell surface, which the immune system could notice are foreign. Most of these peptides are going to be different between the 2 cancers. But what if we can find 2 peptides which are both mutated in the same way? So there's a peptide on both cancers which we don't find anywhere else in the body. If we can find that peptide, we can have a single vaccine to help attack both cancers. And this is really what our lab is going for. I'm really looking at now, what peptides are Mhc Class 1 actually binding in DFT1 and DFT2? Are any of these peptides mutated in either DFT1 or DFT2? And kind of finally, are any of these peptides that are mutated actually present on both cancers? Because if we can find those, we have the foundation for a vaccine that could help the devil survive both of these unusual and aggressive cancers. This slide's just a repeat. So yeah, that's a very whistle-stop tour of my PhD. And some nice pictures of Tasmanian devils to end it. But yeah, thanks to my lab at Southampton, our collaborators in Monash in Melbourne, and my funders. If you have any questions?
Tell me that when you've got the immune system to recognise a foreign peptide and it attacks those cells, how successful is it in attacking enough cells to make the recipient healthy? Or sometimes it doesn't make them healthy, there's not enough?
Yeah, sometimes it probably wouldn't. It kind of depends. We don't really know yet. There's a lot of factors involved. There are some peptides which just don't really elicit much of an immune response. And it depends on a lot of things. But that's something you need to, that's another factor that's quite complicated in the generation of these vaccines. Because you find some peptides which are really unique, they're nowhere else in the body, but for some reason the immune system just won't recognise them. Or it will, but the response is kind of pathetic. But then there's some that just have a huge, huge response to this peptide. So it's completely variable. If we find a peptide that happens to be really immunogenic, then theoretically you wouldn't need that much to get the immune system really going. But more likely it's not going to be very immunogenic, which is probably how they're spreading. So it would be a case of having to use adjuvants and things that can enhance the immune response to the vaccine in the first place. But yeah, it's variable, is the answer.
Sorry, so you haven't got enough knowledge as to why some peptides don't bring about the response in the immune system and others do. You don't know.
So there's a little bit. Sometimes it seems a bit random. Certain amino acids, so ones that have big side chains, that have like rings in them essentially, they tend to have a bigger immune response. Why that happens, I'm not entirely sure, and I don't know if anyone is. But yeah, so bigger, bulkier peptides tend to encourage the kind of activate the immune system a bit more than small peptides. But there's no guarantee of finding the big ones, because presumably that's the first thing that they'll try and the cancers will get rid of as they evolve.
You'd really need T cells from the animals to start to then screen. So if you could isolate some of the immune cells, and then you can screen those against the peptides to see if you're getting a good immune response or not.
Right.
Yeah.
Right.
You don't use radio tracking to zero in on repeat samplings in the wild.
They have just started doing that, haven't they? As it says it's doing
There's
that.
some problem why they haven't before?
I think it's money. And is it the shape of their necks makes them like, they're kind of squat and they don't really have, it's quite easy for them to fall off.
They tend to lose the collars.
Yeah.
Because they don't have a
They don't have a neck.
They've got such a strong bite, perhaps there's no human-made material that can survive.
Yeah, so they have done some radio collar stuff, but I think, it is a bit cost prohibitive, so they've sort of saved it for particular studies where it's been really needed.
So is there any devils at Marble or any zoological structures in this country?
Are there any? I don't think there's any in the UK, no.
No,
There's not that many outside of Australia.
No,
I know San Diego has some.
Yes, and Denmark.
Yeah, but
Copenhagen Zoo has some.
it's quite like few and far between. They're predominantly in Australia.
So how did you get involved? I mean, you haven't got an Australian accent, you've got Australian...
She does.
I was actually born here, but I did all my education in Australia, and at the end of my PhD I was working on MHC, and then I had come across this cancer and became interested in it at that point. So we're quite lucky that we have really good collaborators in Tasmania who are doing a lot of the ecological studies that help with this.
What about the history aspect, the origins of these DFT structures? I understand genetics of an island effect to start with, Tasmania's not very big. And is there any other human involvement other than trying to eradicate them? I think pollution or something. Is there some other element coming in that's not on track down yet?
They've looked for it. So a lab we work with in Cambridge has looked quite extensively for specific signatures in the genome that would suggest, you know, this is common from UV damage or this is common from... they don't find any of that. Well, actually, when you look at it, the most common kind of signature in the genome of the cancers is age-related. So, but it appears in quite young devils. So it seems to be like naturally appearing rather than any external source.
I mean, to me as an outsider, it seems such a mysterious subject that
One thing
really need to nail down what the origin is.
One thing that's still a possibility is viral. So it certainly doesn't spread now by a virus because people have looked really quite carefully to see if they can see any evidence of a virus, but there's nothing. But that doesn't mean that the original transformation was not due to a virus, or even the first transmission events might have been because of a virus as well, and that the traces of those viruses are now lost and we just don't see them anymore. I think that's, especially with Rachel's work showing that it's similar cell types, to me then that makes more sense, because perhaps it's a viral that a virus that infects, I mean, like herpes viruses and cold viruses infecting humans. It could be a similar sort of virus that infects these types of cells.
But marsupials, is there something in the biology of marsupials that gives them a bias towards these sort of more exotic illnesses?
But none of the other marsupials have anything like this. It's strange. I mean, maybe they do, we've just not noticed it yet. I think part of it is going to be that the devil is, I guess, a bit unique in that it has a very easy method of transmission. They fight so regularly, and that's just part of their behaviour. That maybe there's these sorts of cancers are forming in lots of species, but because they don't interact with each other in that same way, we're not seeing it spread. They just kind of form and then die in that one animal. Whereas just because devils, it's obviously got to the face, and then because they bite each other, it just stays spreading between them. So it could just be that. Maybe lots of things are prone to these.
So in that sense, you could have a sort of feedback mechanism going on, which always mystified me with cancer generally, that how does cancer know how to defeat all these mechanisms that are built into you to defeat them? Because it's just a one-off occurrence in a body, and then the body dies. There's no transfer to other human hosts or any other. It's purely down to random, then, isn't it? But if you've got a feedback process, it can accelerate things, presumably. Spreading around through a whole population.
Yeah, I mean, in some ways, there is a similarity to human cancer, or single organism cancers, which will metastasize to another organ. So the cancer starts in one place, and the cells move to another organ, and then those cells have to then overcome a whole lot of new challenges in that new environment, which in some ways is quite similar to these cancers, which instead of going to a new organ, they just go to a new individual entirely. So it's almost like their metastasis process is happening between individuals rather than between tissue.
It's like an extreme version of metastasis.
But what the impacts of that are on the evolutionary rate, perhaps, of the tumour, is an interesting question, whether it speeds up the evolution or...
The marine analogues for these contagious cancers, that's just co-evolution, it's just a random event, there's no chance of crossover there.
Well, other than the one that now exists in a different species, I think that is just it. They've all just emerged separately, haven't they? There's no evidence that they've come from the same. There's been 2 papers on it, and they don't even seem to be really related to each other. There seems to be different mechanisms of how these ones have become transmissible, and they seem to have formed in different ways. So again, they all seem to have emerged separately. So it looks like the emergence of contagious cancers, at least in the sea, might be quite common. Maybe it is something that happens a lot.
Yeah, I think people only really noticed when there was a big die-off in species of bivalves off North America, and they were becoming a little bit concerned. And when they started looking more closely, they found these contagious cancers. But I don't think before that anyone really knew anything.
People were aware that cancers existed in them, but they weren't they had no idea it was.
No.
Was that geographically isolated, or was it all over the world?
When they first noticed it off North America, they thought it was geographically isolated, but now actually they're finding these in
They're in the
lots of
Med,
different-
aren't they?
Yeah, in the Mediterranean as well, off Spain. So yeah, and I think actually they're starting to now look back at where they've had die-offs of particular bivalves and connecting them to
Yes.
cancer. This is sort of joining the dots and going, oh, maybe this was the cause.
Your vaccine, your proposed vaccine you do to the animal, it only protects that individual animal, doesn't it? It doesn't bring anything to their offspring or whatever. And so that would mean you'd have to have a really massive vaccination campaign to make any sort of significant difference, wouldn't it?
I guess it would be a combination between trying to capture and vaccinate ones that are in the wild, but also as these ones that are on the islands and the captive ones are re-released, if they were all vaccinated, it would take a while, but I guess the idea is eventually you get herd immunity to the point where the cancers can't spread anymore. But yeah, it would be, if we get to the point where we have the vaccine, that's going to be a pretty massive undertaking. That would be quite a lot of money probably to get that running.
The government in Tasmania is quite interested in it though. So they've done some re-releases in the north of Tasmania, and they've been quite willing to trial this idea of vaccinating with just the whole cells, the whole MHC positive cells. So that's been quite good. The problem with it is one that it's really expensive to do a vaccine in that way. It's not really feasible for a wild population. And also, we haven't had a really good way then of monitoring those animals after they've been vaccinated and released, so we can't do a proper scientific study in that way. So, yeah, we need other ways. There will be more vaccine trials that will be captive rather than...
Also, that trick with where it doesn't have the code, I don't know the terms, but there's no code there to... Wouldn't that work with, for example, humans? If there was a similar mutation where there was no code, wouldn't that be totally vulnerable to it?
That's really, really common in human cancers. So there's a lot that, because obviously, like I mentioned, how they put up a mutated peptide. Cancers are aware that they do that, but that becomes an immune pressure. And so in human cancers, quite a lot. And it generally correlates with more aggressive cancers. They tend to get rid of Mhc Class 1 on their cell surface. So they just stop expressing it. Yeah. And then they're really hard to treat.
Yeah, there's quite a lot of interest in finding ways in human cancers of them bringing up that Mhc Class 1. In the past, when people have tried like the inflammatory cytokines that we're working with, it can actually cause really terrible immune reactions and what they call like a cytokine storm in patients, and the immune system goes into overdrive and the person can die. So it's not really seen as like a very viable treatment in humans, unfortunately. But finding other ways of manipulating cancer cells so that they upregulate Mhc Class 1 is something that a lot of people are looking into.
I don't know anything about the geography of Tasmania. It's the rivers and mountain ranges that can isolate communities, so you can reintroduce, knowing that it's very unlikely to cross-connect with the community that's in another area.
Yeah, the main one that we know of is in central Tasmania, I think.
So I assume they're free to roam wherever they want to.
Yeah.
There's like a mountain range up there, isn't there?
Yes.
Kind of right through the middle.
Yeah, and it was thought at one point that as the disease sort of spread from east to west, that mountain range would perhaps stop the spread of DFT1, but it didn't. just kept spreading.
So they got over. They're pretty good at getting over.
It's not birds as a vector connecting the two or something like that.
It's a really interesting idea, actually.
There's no reason it couldn't be. I don't think. I think they just kind of assumed that the devils had got up there.
Yeah, but that's, I mean, like we don't really know, I guess, how long the cells can live outside of the devil. I mean, if they were in a carcass or presumably they would be a little bit like a virus. They could live for a little bit of time before they were picked up again.
I'm also thinking of the business over here where there's government's trying to suppress badgers because of bovine TB in farm animals and that. And the theory goes that you suppress them, kill them off in one area, then others from outside move in with the a TB, you're back to square one sort of thing. That sort of human manipulation doesn't always work. It's one how you do introduce into an area without disturbing what otherwise might naturally be going on.
I'm not sure how do they do the re-releases? Is it?
Yeah, I don't think they worry too much about.
I think they just want more devils back on the island.
Yeah, I'm not sure there's that much. I mean, when you look at the amount of analysis that's done on the badgers, I don't think that level is actually done in Tasmania, unfortunately, and how, in releasing these animals.
So how many are around at the moment?
In Tasmania?
In the wild?
I think about 20 or 30,000. To give you an idea,
it was 150,000 before 96.
Yeah, well, between 120 and 100, like the.
And the last surviving one in mainland Australia would have been.
So they think there's a bit of debate about when they went extinct on mainland, but I think general consensus is probably about 400 years ago. So it was quite shortly after humans settled and dingoes and things. Yeah.
Have they ever become pets with the people in Tasmania?
Don't think so. Apparently they're very nice and nice-natured until they hit about 2, when they hit sexual maturity, and then they just become a nightmare. They get suddenly. So I mean, a good explanation of that. I mean, the difference between this young devil. So this, you could probably go up and pet this. It's a young devil. They're generally quite nice, quite placid. You see that, any places, rescue centres and sanctuaries for devils, you always see the people who work there handling the babies, but then as soon as they hit about 2, they're suddenly doing this, and they look a lot more aggressive, and they start biting as that's kind of just how they interact. So I don't think you'd really ever want one as a pet. Apparently they smell terrible as well.
Trying to find pictures for this poster put in libraries. Very, very difficult trying to find pictures of someone snarling.
Oh yeah, no, they're good ones. I always go for the baby ones because they do look really cute.
But I was saying,
there was a huge change between when they're young and when they suddenly get quite aggressive. So I don't think they'd be suitable pets, because if they did catch you, they could do quite a bit of damage.
Yeah.
I mean, Australia's a massive place, but I always think of it as where anything can go wrong with human interactions, it does go wrong. I think the mice, the rabbits, the cane toads, just don't get anywhere. I'm just wondering if it's even worse case in Tasmania.
Well, I mean, there's always problems with stuff like that on island populations, just because they generate such a specific ecosystem that becomes very finely tuned, and it's quite a delicate balance of everything that's going on. And when there's nothing to kind of come in and balance it out again, if you're limited to what is on the island, the second you bring something in, it can all go. There's no way for the ecosystem to really fight back. Whereas if you've got you look at mainland Europe, for example, it doesn't happen on such an intense scale, because there's natural stuff that can then come back in and refill the gaps. If you accidentally wipe out something on Tasmania, there's nothing to come and fill it in. It's isolated from everywhere. So you do see a really, and Australia gets it because it's also a very big island, but I mean we see it here, things get wiped out quite quickly by small changes in the environment, just because there's not really anything to come in and, it's all very
the UK
delicate
has a lot of
balance.
the same issues, doesn't it? And the idea that, things are on a lot of the islands offshore in the UK, like the red squirrels. They're a really good example. And so some of the only populations in the south are now just on the islands, and it's a dangerous, way to precarious.
If anything gets onto St. Ramsey Island, they're gone.
Yeah.
One grey squirrel gets over there, and suddenly.
Yeah, and they have disease issues as well. So I think it's sort of similar problems actually.
Yeah. The island effect.
Yeah.
But that's more of genetics. Well, I suppose it can be genetics and climate and things like that.
Yeah, I think it affects it from more ways than you maybe initially expect. Kind of being isolated on an island over time obviously massively affects the genetics, but it is just little things like the fact that there is nothing else that can come in and replace it. So one thing goes and suddenly the entire island has to change. Or one thing comes in and everything, they're really responsive, I guess, to outside interference.
So what made you want to do the Tasmanian devil for your PhD?
So I was always really interested in cancer at uni. I'm a biochemist. And so I wanted to go down the cancer route, but the reason I actually went into science was because I always loved like nature. I wanted to be a vet when I was a lot younger. And then decided I didn't really like the people-facing side of it so much. Didn't want to do like the operations and things. So I went into science. Then I kind of put the idea of doing work on animals out of my mind and went down the cancer route. And while I was looking for PhDs in cancer, this one popped up, and I didn't realise PhDs existed that had both endangered species and interesting animals and cancer in them at the same time. So it was kind of like a perfect role came up for me. So yeah, I mean, it was just, you kind of apply at PhD level, you apply to a specific project, and this was just a perfect one for me. So I applied, and thankfully I got it.
Yeah. It does seem a bit strange. I mean, it's solely on Tasmania, and there's not just you, but a section of a department at the university, the other side of the world, heavily involved with research on this.
And there's another lab in Cambridge as well, in the UK. So there's a few labs in the UK that are working on this. And is there one in America, or is it just an American guy in Tasmania?
No, there's a lab in America that does work on it as well. Yeah.
So there's a few like.
So is that fairly common? I'm not talking about Tasmanian devils, but that structure within academic biological.
I think it comes from the fact that people are generally will have an underlying interest in a biological process or molecule, and they end up studying a pathology or disease that allows them to kind of study that molecule, if that makes sense. So, I mean, I guess I came into this first with an interest in Mhc molecules, which are found in all vertebrates, they're everywhere, and involved in a lot of different pathologies, and then sort of became interested in this. And we still have interests in other areas as well. And I mean, the group in Cambridge, I guess, they're interested in tumour evolution. So they use this as a model for understanding tumour evolution. I guess in a similar way, although we have a very definite that we want to do something, it has to be a practical outcome of the research in terms of, saving a species. But we're also trying to get at what are the underlying mechanisms that allow tumours to either be recognised by the immune system or ignored by the immune system. So I think that's why, the group in America, they're interested in modelling how diseases spread and how different factors then influence the rate of transmission and so on. So I think it's a combination of people seeing something that is a real issue at the moment and a problem that they can sort of address, and kind of having a scientific interest in something that's a little bit more broad, I guess.
I suppose 20 or 30 years ago, you couldn't have done this transnational research. The principle of cell line development wasn't around, so you'd have to physically go where the problem was, to Tasmania. That's something
that's right. That's true, actually.
Yeah, the fact that cell line stuff is so kind of, it's been well
and
developed now.
it's totally immortal that you can come back 50 years' time and it'd be still the same cell line.
Depends how long. If you keep freezing them when you're not actually growing them, then you can keep them reasonably stable. But they degenerate, they change. So the most famous cell line is a HeLa cell, which you might have heard of. It's a cervical cancer cell from a woman in I can't remember, was it the
Yeah,
60s?
yeah.
It was a long, long time
Oh,
ago.
Arabian woman.
Yeah.
Yeah.
And they,
Yeah.
so everyone grows HeLa cells, but nowadays, if you actually look at them, they have an obscene amount of chromosomes that have appeared from nowhere. They don't really have half the organelles you'd expect to see in a cell. Like, it's kind of at the point where now people are questioning, are these even really cells? Like, are they a model of cells anymore? So you do have, there are limitations. And we even see, so we bring them up from frozen and you grow them on the plate, but when they fill up the whole plate, you have to take them off and then put less of them on and you put them into more plates and grow more. But every time you do that, like, there's only a certain number of times you can do that before you actually see the cells start changing. So we see...
That's not just these HeLa cells, this is...
No, this is also...
general cell line.
So this is in DFT2, which normally look like... I'm just trying to find the slide. So they normally look like this, which you can't really see it that well. They're... So they kind of look like nerve cells. They're like, they have a little nucleus. Yeah, it's not a great picture, but they like spread out, so they almost look like stars. So they have lots of little projections that stick out. But actually, the longer you grow them, those start to, like, they get shorter, they start growing strangely, they stick to each other. They do all sorts of weird things when they get quite old. And so you can actually fit, even without looking at what's happening, like looking at the genes or the proteins or whatever, you just see the cells start to look different after you've been growing them for too
It's
long.
like our telomeres.
Yeah.
So telomeres.
So yeah, you do have to be a bit careful with them. So that's why we have multiple cell lines, really, to kind of confirm that we guess they've all been isolated from different devils and different tumours at different times. But I guess the idea is if they're all still reflecting each other, and then we can also reflect the tumour samples with it, then they're still a good enough model for us to use. But you've got to be careful, there's a lot of limitations with cell line work. They've revolutionised what we can look at, but they are limited.
Well, you've got a super job, that's for sure.
It's interesting, I do enjoy it.
Any more questions? No, I think, what's the time? 10 past 9? Well, thank you.
Yeah, wonderful. Thank you very much.
Thank you. Thanks for having me.
Very, very interesting.
Oh, thank you. Thank you.
I think you'll find Mark Craig's coming in the next
couple of months.
Yeah, so that'll be like another, that's a very human, clinical side that I guess we'll be talking about.
Yeah, it'll be a lot of the same principles of
Yeah.
immunity and